Bioinformatics for Precision Medicine. Hello, everyone. My name is Mohit Mazumdar, and I'm one of the co-project mentor of the program. So, as for today, let us go through the sessions that we are going to have, what actually they are discussed, and what we have in the curriculum, how that aligns with the subject and the importance of what we discussed today. So let us go through the sessions as one by one. One second. Yeah. So the first session of the program will be on 6th July, and it will start with an overview of all the resources that we discussed, and also the access to the program. So for the first session, this will include program orientation, group discussion, to learn from the participants of the program and about the research interest and objectives. So the orientation is also is to get you familiarized with the introduction to bioinformatics. So followed by this, the first session, which is a scheduled session which focuses on the problem of cancer. So in this session, we will also discuss about the complexity of cancer biology and the typical challenges that you see in diagnostics at organ, tissue, or cellular level. And we will also discuss about molecular factors that are involved in tumor development, that are there in for basic and both translational research in oncology. So in this study, where we have also few examples, like Ilya said to all of you, that we have already prepared a few examples. So in one of the examples is a study where you can see on the screen a patient derived cell line, and this in this the xenograft models are also used in another study. And we have also data sets where we use clinical trials information that offers a collection of data to understand and get information about the diagnostics and also treatment of cancer in patients. So the next session after that, which would be the second session. In the second session, we will talk about the technology after getting an overview of cancer and how deadly it is and what are the different characteristics to understand a cancer cell. So in the second session, it will be more about the technology, as I'm saying. So it would be about understanding how next generation sequencing works. You will learn about shotgun sequencing reads, and then you'll learn about how to make the data structured, and then you will learn about different bioinformatics methods that uses like processing, where we will be doing both germline and somatic mutations. We'll look at the analysis, where we will do mapping, detection, variants you can see on the screen, and also in this in the last part. So in the last part of the session, we will discuss about the interpretation. So how can you interpret this data? And along with this, like you will have access to the resources that explains all of this, so that you kind of get familiarized with what is being covered and have a chance to do it at at your own pace. So anytime that you feel that you have time to go through the resources, these are online courses that are available on the Learn OmicsLogic portal. So in here, I mean, we have different resources that it starts from a very basic introduction, and then goes into different processing pipelines and how data can be interpreted and how you can structure that data. That could make sense because it's from a large number of it's a big data, and then you're transforming into a very meaningful data set. So in this study, in this session, I mean, we will learn about how genomics is used in biomedical drug discovery, and how genomics is used in different other areas in translational research. So in the third session, which will be on processing of another type of sequencing data, which is the transcriptomics data. So in this session, after learning about the genomic processing data and its application in biomedical research and precision medicine, you will learn how we can measure functional proteins and look at the mRNA abundance, and accurately understand the alternative splicing. So you saw examples of RNA sequencing before. So going through this entire process of RNA sequencing, doing the read quality and pre-processing yourself, going and then mapping it to the reference genome, and then understanding its role by quantifying the genes and also the transcripts. And in the same way, after doing that, you would want to do the visualization. So I think this entire process is also to let the scientific community know of what you have what you have obtained as a result. So expressing that in a visual form and getting the visualization is a very important part of our scientific things that we do every day. So in that sense, we will learn about how you can correctly and visualize the gene expression information, and also do PCA to do that, and also go through annotation and interpretation. So in this session, we will be going through for example, the processing pipelines. We will prepare a full pipeline and then go through different type of analysis that we do for gene expression data. So the session after that, which will be the fourth session, is on machine learning. So you have already learned about two overview of two different technologies, which is genomics and transcriptomics. And then you would want to learn about how do you do this, make sense of this data, and do this to find out different patterns that are lying within a large number of other data sets also. So finding predictive modeling, or like understanding how what this data have information. So this complex collection, where this is about the data and their complex collection, we are looking at different types of characteristics. We are looking between correlation between clinical manifestation of disease, molecular patterns, and how we can define it using different statistical analysis, right? So machine learning approach allows computational power that could be applied to these challenges. So in this session, we will use machine learning. And during this time, as I was saying, that there will be resources allocated that will help you go through and do this on your own. So you will also have access to the coding platform, coding playground, which is there in Learn, where you can write and do scripting in Python and R. And these also they are also like tutorials that takes you through this entire process. So you would know how to apply R and Python in your genomic or transcriptomics data analysis. So yeah, that would be an experience, and that those will be available to you as the session goes on, and we will cover many of those aspects in these live sessions. So after going through and learning about how do you visualize and how can you actually analyze from different project examples that we have prepared for you, the next thing is to go and do it yourself, like finding those important repositories on, and finding the data sets from those important repositories like SRA, Geo, NCBI, that are there in NCBI. And Eli already discussed about the TCGA repository. So this session will be about those different repositories that are there and very important for any bioinformatician to explore and understand how to extract the information that you need for your project. So we will be discussing in that respect. And about the opportunity, many of us here do not even understand the opportunity that is related to training like this. Why we are doing this, why we are saying that independent research is now very much possible and within reach of everyone. Why is so? It's because the availability of data. And yes, that kind of data is available. So and NCBI is such a good example of that. You can see that how many data sets have been already deposited, and on the way it is growing rapidly every day. So after learning about the data sets and how to figure out and find data, we will gain after the first phase. I think it would be 5 sessions, and then after the fifth session, the sixth session would be about next generation sequencing, where we will learn about viral genomes in the host transcriptome. So here we will also learn about oncolytic viruses. So that is one of the um, feature of learning through precision medicine program is to learn about different aspects of transmissions from cancer and then these different viruses. So here we will also learn about like how they are involved in tumor development. How do you connect from virology to cancer, and then learn about inflammation and tissue damage. Here we will also learn about how can we align, annotate, and do uh, you know mapping. So we will learn about viral genomes, and we will also learn about alignment to databases of those viral genomes. So after learning about that, so the second phase in the where we are going to learn about different aspects of how can we interpret virological data from for virology and infectious diseases. So in this session which is session 7th, we will learn about multiple sequence alignment and phylogeny. So here we will learn about how can you study viral genomes for liver, blood, and neural tissues. And we will also learn to prepare data and setting up bioinformatics pipelines, where you will find to do phylogenetic analysis and multiple sequence alignment of these genomes. So I think most of you are familiar with phylogenetic tree. So phylogenetics is a study. Phylogenetics, in the study of phylogenetics, you learn about the lineages between different species and how you can interpret that. So after that, so which is the 7th session, so this is spanned across 2 sessions a week. So I think it would be all right, and we'll be going through this, and you'll get enough time to complete the assignments and also go through some of the courses and materials and data sets that we have prepared for you. So learning about that, then practicing. So it's a very important aspect of this program is to do the hands-on tutorials and do the hands-on practice yourself to gain the most of it and become an expert, right? So to do that, you have to be more comfortable by in the on the platform that you will have access to during the 1 month program. So here we will learn about workflows and pipelines in bioinformatics. So we have discussed already. I've seen a lot of pipelines and workflows. They are like algorithms that are put together in a logical manner, right? So and that in that way, we have technology that helps us to find out and detect those challenges and find out critical important information to design to design our research correctly. So this is a very important role that plays in bioinformatics, and here we will learn about detecting and mapping those to databases, and we will also learn about how you can understand about how genomics and genomes and the evolutionary time correlates. So this is something that we have planned for this session, and here we will also go through some of the processes in this. This is another one of the example. This is a birth death example of evolutionary study. So here you will also, there are like parameters that we will cover that are there to understand this sort of algorithm, and what are different algorithms to do this and how they can be applied for a problem. So about the problem, so now the next session is about the problem from infection to pandemic, from a viral adaptation. So here we will have an example about the coronavirus pandemic, and we will do takeout sequences from NCBI virus, and we will do comparative studies where we will compare these 4 sequences, genomic sequences, full sequences, and find out like how they can correlate and how they make sense in activation of immune response, and then cytokine storm, which is caused by viral replication, and also could relate to what is happening currently um, everywhere. So as I said, that you would be able to do it alongside with us. So we will use the TBA Info platform, and where we have sections on to do this, which is virus quasi species. So we'll discuss this and we'll tell you like how this is done um, in session 9. So in after session 9, the last session, where this entire story comes to an conclusion, where we are going to learn about host pathogen interaction. So after applying and learning about all of these technologies and how they can be done for different diseases, sorry, here we are going to learn about what are the strategies that we can apply to overcome. So we will talk about vaccine design, so how bioinformatics, or how you can consider data and then think about vaccine design. So you have a lot of information coming from big data, you know about the sequences, you know about the gene expression. Now you have figured out that this is a protein of my interest, and I think these are the possible sites, and based on the mutations that I've seen in the genomics, these are some of the places that we should target, and that these are some of the epitope binding sites. So based on all that information, the important thing is to go for a data driven hypothesis, and that data driven hypothesis is something that we are uh, going to discuss throughout the program. So in the session, we will cover about some of the structures. How do you connect from uh, host response to viral replication, and then implications of that to cell, organ, and tissue. So exploring different viral strategies for exploitation of host machinery and the viral protein production. So these are some of the topics and sessions that are there for precision medicine. And I think one of the best part of this is that we also will be doing giving and going through the coursework, which actually is pretty amazing, and that's what we have heard from all of our participants so far. So after doing this, and if you have any confusion, and if you think that bioinformatics is difficult, because we are integrating so many things, we are integrating data, we are integrating algorithms, we are integrating complex machine learning methods, it sometimes gets like really thought it to be like, sometimes it gets really challenging. But we are here to help you out with that, because we have gone through this entire process over and over again, and thought of different strategies that actually works. So